So what I'd like to talk about is mitochondria and particularly how they impact us as we get older. But kind of starting at the beginning, kind of very briefly, what are mitochondria and why are they so important to our health?
Yeah, mitochondria are little organelles we have in the cell, multitudes per cell of these small organelles, and they have many functions. For a long time, people thought they were just making energy in the cell, and that's one of their most important functions. But they also participate in multitudes of other processes in the cell, interact with the environment in the cell, with the nuclear processes. So the mitochondria are not part of what we call the nucleus of the cell. They are around the nucleus, they're in the cytoplasm of the cell, but they play enormous amounts of important roles in processes, also in aging. And so with aging, we know that there's a loss of mitochondria. There's also a loss of function in the mitochondria. And we have begun to target mitochondrial dysfunction as a very early indicator of neurodegeneration. So that is one of the things that really attracts my interest, is that it seems to be one of the earliest things that happen in the cell is some changes with mitochondrial function that then leads, are the early steps probably in Alzheimer's and other neurodegenerations. So it's very important to understand these changes at the earliest possible stage.
So how do you tell that the mitochondria, is there any kind of clinical way of telling that your mitochondria are not functioning correctly?
Yeah, there are a number of different ways. And mitochondria, by the way, are very heterogeneous. We have different kinds of mitochondria, different sizes of mitochondria, different functions of mitochondria, even within the same cell. So it makes it quite complicated. And then the genetics rely on the female rather than the male, as is well known. And so the whole organization and genetics and of mitochondria is very intriguing. And while in the nucleus, we have a large genome with 20,000 genes and very complex structure and organization, the genome that is found inside the mitochondria is a very simple, small plasmid or circular DNA with only few genes that code for mitochondrial energy function. And other proteins, other functions that are being brought into the mitochondria are coded for by the nuclear DNA. So we have two different genomes within one cell. It can be the mitochondrial genome and the nuclear genome.
So how do mitochondria change as they get old, as we get old? So we talk about the dysfunction. In what way are they dysfunctioning?
Yeah, so you also mentioned how do we measure mitochondria? So we can measure mitochondrial function directly with energy measurements in sophisticated equipment. And we can also look at the mitochondria with electron microscopy and look at the structure of the mitochondria, organization of mitochondria, how many mitochondria there are, and other such things. So what's been observed is that there are a number of things that go wrong with mitochondria with aging. Energy production can change, usually go down, but can also be activated. So it gets dysfunctional. It gets out of wacko with age in the mitochondrial energy production. And then there are other processes in the mitochondria. We can also observe the structure of the mitochondria changes. The fusion, fission, we call it, between how mitochondria organize themselves is also changed. So there are a number of processes that change with aging, and also early on with neurodegeneration, for example, and other diseases.
Yeah, I want to talk about the disease, but one thought, so the mitochondria, they regenerate themselves, right? They separate.
Yeah, fusion and fission, yeah.
And fission. I mean, if they're generating new ones, why are the new ones kind of older? Can't they generate new ones which are younger?
Yeah, it's an interesting question. Yeah, they could in a way, but they really can't. So it depends on a number of things, also the genetic background of mitochondria and many processes. But we have a heterogeneous population of mitochondria. So there can be mitochondria at different, should we say, stages and ages within one cell.
Mm.
And that makes it complicated. But mitochondria can also be disposed of and new mitochondria generated all the time.
So I was going to ask about that. So mitophagy and the getting rid of the existing mitochondria, which seems to be a way of helping with their health. Like, how does mitophagy help with the overall function of?
So mitochondria get bad with aging and neurodegeneration, as we just talked about, in different ways. And the process of replacing these mitochondria, getting rid of the bad ones, is the mitophagy process. So we have a general process called autophagy in the cells, getting rid of cells, more general ways. And then we have a targeted one that gets rid of the bad mitochondria. And you can actually tell that mitochondria are bad, if you will, or dysfunctional in for example, electron microscopy, where the structure looks abnormal, and it just simply, so they need to be disposed of. If we don't get rid of those bad mitochondria, they accumulate, they fill up and take and simply cause various problems in the cell, the bad mitochondria. So we really need to have an effective process of replacing the bad mitochondria, and that is done by a special organization involving lysosomes and acidic milieu in the cell that then degenerates the mitochondria that are not functional.
Let me start with the mitochondria. So our body is made of cells. All cells, except perhaps red blood cells, contain mitochondria. Mitochondria have multiple roles. They're the energy source of the cells. They also have function as immune regulators, as metabolic regulators, as inducers of programmed cell death. And mitochondrial dysfunction is involved in many different diseases, but particularly in diseases of aging. And everything we said about aging in general applies to the mitochondria in spades. Mitochondria are much more prone to DNA damage. Their function declines much more rapidly than nuclear function, and they accumulate neuromutations and see damage as we age. The one thing that makes the mitochondria unique is it contains its own chromosome. This chromosome is much smaller than the nuclear chromosomes. It's only 16,000 base pairs, which isn't much, and it contains 13 large protein-coding genes that are part of the so-called respiratory chain that allows the mitochondria to turn oxygen into ATP, and also has its own ribosome system that allows it to produce proteins within the mitochondria. That was the thinking up till 20 years ago, when we and a couple of other groups stumbled on an observation that there is a small gene within a gene in the mitochondrial DNA that a scientist who has already passed away from Tokyo, Japan, named Nishimoto, he named that protein humanin. My lab cloned it at the same time that he cloned humanin. He was the first to recognize its importance and publish it. We became friends in the early 2000s, and unfortunately, he died from cancer a little over 10 years ago. Humanin turned out to be this novel small peptide encoded in the mitochondrial DNA. We believe it gets the mRNA for humanin goes from the mitochondria to the cytoplasm, where it gets translated into this little peptide, 24 amino acid peptide, gets secreted out of the cell. and then circulates around the body and has really tremendous beneficial effects, from protecting the brain from cognitive decline, from protecting aging tissues from fibrosis and other damage, from delaying or preventing atherosclerosis. And there are now probably over 500 publications from over 30 laboratories around the world demonstrated the various beneficial effects of humanin in animal models, mainly mice and rats, in primarily the context of aging. And so humanin is sort of the quintessential mitochondrial peptide. So about 17 years ago, 15 years ago, in one of our lab meetings, we started asking, if there is one small open reading frame hiding inside a larger gene, why shouldn't there be more of them? And we started studying the mitochondrial DNA, looking for additional open reading frames. And by now, we realized that there are many hundreds of them, probably 600 or 700 small open reading frames, each of which can be conceptually translated into a small peptide. By the way, these days, we like to call them microproteins rather than micropeptides. That's the latest trend. And many, many hundreds of microproteins can theoretically be made by the mitochondrial DNA. We have now believed that we identified dozens of these in the mitochondria. And the big change that has happened in the field is that people have started recognizing that there are microproteins, these small open reading frames that get translated into peptides. They exist also in the nuclear genome. And there are actually hundreds of thousands, if not millions, of small open reading frames that can encode microproteins in the nuclear genomes. And there are multiple groups working on these. In fact, there was this really interesting family of molecules known as long non-coding RNAs that was discovered about 15 to 20 years ago. And by their name, it implies that they don't code for any protein. Now people are recognizing that many of these long non-coding RNAs actually do code for microprotein. So the science of microproteins is expanding. We like to believe that we started it and that we really are moving the needle in the mitochondrial sphere, but many outstanding scientists are now working on nuclearly encoded microproteins that have important biological roles. I happen to believe that these microproteins from the mitochondria, from the mitochondrial DNA that we work on, are particularly important in aging, precisely because during the aging process, there is so much damage to mitochondria and mitochondrial DNA that the production of these mitochondrial microproteins goes down, leading to poor performance of certain things, certain tissues, certain biological processes, and resulting in diseases like diabetes and Alzheimer's disease and other diseases like fibrosis, which mitochondrial microproteins can repair, correct, and reverse. So when we recognize that there could be more mitochondrial microproteins, we started out initially by just saying, okay, here's humanin. Right next to it, there's another open reading frame. Let's see what that one does. And we named a bunch of them small humanin-like peptides, or SHLEPs. One of them is called SHLEP2, and it's a very exciting microprotein that we're working on, been published now in about 10 or 15 papers. And to me, it's one of the most promising candidates that can be translated into human disease, and whether that disease would be Parkinson or some other condition, we're still unraveling. But about a decade ago, and we published this in 2015, we noted that there was a small open reading frame a little bit further away from humanin, which we called MOTC. And MOTC turned out to be a fascinating microprotein. And it has exercise mimetic and exercise enhancing biological activity that result in weight loss in mice that are obese or fed a high fat diet, reduction in steatosis, or hepatosteatosis is fatty liver. One of my colleagues is now showing that it also decreased something called myosteatosis, which is a really underappreciated severe process of aging where there's fat accumulation in muscle. And MOTS-e ameliorate that and reverses that. So MOTS-e, which is also regulated by exercise, if you exercise, you secrete MOTS-e, is an exciting peptide with potential roles in a number of conditions, from frailty to obesity to fatty liver. And we licensed this to the company Cobar that Nir Barzilai and myself founded a little over a decade ago. And it has completed phase 1 studies and is now planning phase 2 studies with MOTS-e, as well as identifying several other potentially important microproteins from the mitochondria, which they will hopefully develop for additional diseases of aging, such as idiopathic pulmonary fibrosis and related conditions.
Do you know magnesium is a critical mineral for cellular function? Perhaps the most important role is being bound to ATP, the currency of cellular energy, as magnesium ATP complex, which is the recognized form of the cofactor required for proper function by hundreds of enzymes. Research suggests that magnesium deficiency can contribute to increased oxidative stress, accelerated cellular aging, and mitochondrial dysfunction. To make sure that we are not deficient and to support optimal mitochondrial function, my wife and I take Magnesium Breakthrough daily. Unlike some supplements offering limited forms, Magnesium Breakthrough is made of all natural ingredients and contains a full spectrum of 7 forms of magnesium to ensure well-rounded support for your cellular health. If you want to boost your mitochondria, simply go to bioptimizers.com/modern and use promo code MODERN10 during checkout to save 10%. Thank you for your support. Do the mitochondria in the different tissues produce? Are they different? I mean, do they work in different ways, and do they produce different peptides?
Absolutely. And I think it's not really appreciated even by many scientists that the mitochondria from, say, brain and liver and muscle are very, very different. They look different by electron microscopy. If you isolate mitochondria and do a proteomic analysis, you see very different distribution of proteomic results. If you look at the function, they're quite different in their energetic profile. So it's no surprise that we identify different profiles of expression of mitochondrial peptides. One of the techniques that we've recently developed is the mitochondrial equivalent of what's known as RNA-seq. RNA-seq is a technique that was developed to measure the RNA from tissue samples and look at all the 20,000 genes that express RNA and eventually proteins and see what goes up and what goes down in various conditions. The standard RNA-seq does not look at microproteins, certainly not in the mitochondria, and we've developed a method to specifically analyze the expression of mitochondrial peptide at the RNA level, and they're very different from tissue to tissue, and it's very sensitive to disease states and interventions of all sorts. And indeed, the profile of mitochondrial peptide that are produced by mitochondria from different tissue is very different.
Interesting. Yeah, because I always thought of mitochondria as being the same, like everywhere.
Kind of like standard issue, you stick them in cells and they just make energy, but it's not the case.
That's interesting. Yes.
There's a concept of mitochondrial transplant, right? So you can take mitochondria and put them into tissues, and taking them from one type of tissue to another can have profound effects. It's still a concept that is in development, and it's not quite ready for even clinical trials. But which mitochondria you start with is going to have a major effect on the results you're going to get.
So one question on mitochondria and aging. So does the DNA damage in a mitochondria just accumulate and accumulate like over our whole lifespan, or do they have like, I don't know, can they reset?
Right. So the mitochondria employ a very different model than normal mammalian cells. They are essentially continue to function the way unicellular bacterial-like organisms do. They divide much more. First of all, a cell has two sets of chromosomes in one cell, and the cell divides as needed in the tissue. But each cell has hundreds, potentially thousands of mitochondria. And the mitochondria divide in an asynchronous fashion with the rest of the cell. And there are many mitochondria, and they undergo a process known as mitophagy, which is kind of like autophagy, where a whole cell gets sort of eliminated when it's done with its role. Poorly functioning or sort of not quite 100% effective mitochondria gets eliminated through the concept of mitophagy. And even though we said that there's a lot of damage that happens to mitochondria as you age, a lot of these less than perfect mitochondria get eliminated through this mitophagy process. So you're always going to have some functioning mitochondria. But if you're, 18, your body is really good at getting rid of all the abnormal mitochondria and keeping like a really good cohort of fully functioning mitochondria. By the time you reach my age, there are more and more of these sort of semi-functional ones that are trying to do the job. By the time you get to 100, there's even less quality control there. And that happens at a steeper rate than some other components of the cell, all of which age, but mitochondria seem to age fastest among the different elements in the body.
How do the mitochondria change with age? And kind of what are the driving factors that push them?
So again, it's a complex matter. I mean, in the sense that mitochondria are very complex and dynamic organelles, and they, let's say, what happens normally, the way that they maintain homeostasis is that they are constantly generated and replaced, essentially, depending on the stimuli and the metabolic inputs that they get and the needs of the cells. So, and again, as I mentioned also before, the way to maintain mitochondrial homeostasis is either through generating new mitochondria, so mitochondrial biogenesis, or mitophagy in order to clear damaged mitochondria. And also mitochondrial dynamics are very important for this, because essentially mitochondria can be either fragmented and become like more isolated and so on. And this is when they, for instance, can be taken up if they are damaged to be degraded by mitophagy, or they can be forming very extensive networks to actually improve or become more efficient in terms of metabolic function. So this normally works very well, and it's very dynamic, and they can adapt very quickly in a youthful or healthy status. However, over time what happens is that due to the decline also, or in the other pathways that take care of things like mitochondrial clearance, loss of, let's say autophagy, or other pathways that should be taking care of reducing the damaged mitochondria, and the fact that also mitochondria function in terms of generation of the energy, so the balance between the different complexes within the mitochondria that would generate ATP becomes altered due to, again, alteration of protein synthesis, reduced mitochondrial biogenesis, and so on, then the mitochondria start becoming also, let's say, less functional in terms of generating energy. They may generate perhaps more ROS and more oxidative stress, and they tend to accumulate also, so because they cannot be removed anymore efficiently through mitophagy during aging due to the decline in this process. And so what happens is that you tend to accumulate dysfunctional mitochondria, which then in turn, they can start generating damage also within the cells, right? Because if you have essentially more oxidative stress and let's say less energy generation and the inability to remove these mitochondria, essentially there will be that's when essentially problems start occurring, because then you get an influence on the other hallmarks as well. So these hallmarks are all connected, right? So that's where it becomes important to maintain homeostasis. Yeah.
So how do we encourage mitophagy?
So there are several genetic pathways that modulate mitophagy. I mean, in terms of essentially that can be also stimulated through, let's say, nutrient sensing. I mean, there is the involvement, for instance, of phosphorylation processes that sense and detect damaged mitochondria, and then they stimulate the ubiquitin proteasome system essentially to remove this. So they tag the damaged mitochondria, and then they recruit the ubiquitin proteasome system in order to get rid of them. Pharmacologically speaking, there is, I would say, people are actively looking into ways to specifically target mitophagy, but I would say at the moment, perhaps one of the molecules that I think it's very well known also in the supplement space and so on is urolithin A. So that one, it's a natural molecule. It's GRAS recognized as generally safe. It's been already used in humans a lot by, essentially it's been discovered in my ex-lab at EPFL in the OREX lab, and is now being used currently by companies to do clinical trials. And that one seems to very strongly promote mitophagy in several tissues, and in particular seems to work pretty well for brain and muscle. So preclinically speaking, with some initial evidence now going for muscle aging in humans. So that one seems the most promising at the moment. Yeah.